we will discuss how the antimicrobial resistance is transmitted from one bacteria to another bacteria. So grossly, transmission of antimicrobial resistance we can divide into vertical transmission and horizontal transmission. Vertical transmission from the parents to progeny, vertical. So one bacteria, when it divide into two, both the bacteria are resistant. So this resistance, the most important mechanism is a mutation. So bacteria develop some mutation, and that mutation is passed on into all the progeny. So all the bacteria become resistant. Now what the mutation can do, what can happen due to mutation that the resistance occurred? So there are many mechanisms of development of resistance. So what are those mechanisms? The bacteria may develop some enzymes, inactivating enzymes, so that can kill the antibiotic. So when the antimicrobial is broken, the bacteria will become resistant. And the development of inactivating enzymes is the major method of drug resistance to three main antimicrobials, remembered as A, B, C. A for aminoglycosides, B for beta-lactams, and C for chloramphenicol. The resistance to these occurs because of security guards we discussed as the example. So aminoglycosides, beta-lactams and chloramphenicol A, B, C resistance occur due to inactivating enzymes. In case of beta-lactams, they are called as beta-lactamases, whereas in other drugs they do not have any specific name. Okay. The another method of development of drug resistance may be the bacteria may change its target where the drug was working. So it may alter its target, and this type of drug resistance is most important in case of MRSA and similarly VRSA. In MRSA, the bacteria change the penicillin binding proteins. Similarly, in VRSA also the target is altered. So altered target may lead to drug resistance. Another method is a decrease in permeability. If the drug is not able to enter inside the bacteria, it will not be effective. This is the major mode in case of aminoglycosides. So enzymatic method is also there, permeability is also there. Then one more method of development of drug resistance is development of efflux pumps. Efflux pumps. So as we discussed, in case of tetracyclines, the bacteria develop efflux pumps and they remove the tetracyclines out of the bacteria. So we developed a drug called as tigecycline, which was resistant to efflux pump, tigecycline. But now some of the bacteria have developed efflux pumps even against a tigecycline. So efflux pump is another method of drug resistance. Then lastly, the bacteria may alter its metabolism. So this method is main with sulfonamides. Remember, sulfonamides, they inhibit the folic acid synthase enzyme. So these drugs were working on the concept that bacteria do not utilize the pre-formed folic acid. So bacteria make their own folic acid. So if we inhibit their formation of folic acid, bacteria cannot grow. But when we give sulfonamide, some of the bacteria start utilizing the pre-formed folic acid. So that can result in drug resistance. So these are the methods of drug resistance. So coming back, how the resistance is transmitted. Vertical transmission is mostly through mutation. So that mutation is transferred to all the progeny. But more important is the horizontal drug transmission. Horizontal drug transmission means one bacteria is resistant, it will make another bacteria resistant, not the progeny. Horizontal, brother sisters. So how this bacteria make other bacteria resistant? So we have three major methods of horizontal transfer of drug resistance. These are conjugation, transduction, and transformation. conjugation, transduction and transformation. We'll try to understand. Suppose this is one bacteria which is already resistant. This is the second bacteria which is not resistant. So first bacteria, it already has a resistance gene. So this resistance gene is already present in the first bacteria. So first of all, this bacteria will multiply its resistance gene. It may be on the plasmid, transposon, at any place. So first of all, it multiply that resistance gene. Many resistance genes are produced. Now this bacteria forms a physical contact with the another bacteria which is not resistant. This physical contact is known as sex pili. Now through this physical contact, the resistance gene is transferred into the second bacteria. So second bacteria also become resistant. So this method is known as conjugation. So conjugation is with the help of sex pili. So one bacteria transfer the resistance to another bacteria through actual physical contact. Conjugation. Then second method, first bacteria has a resistance gene. It has multiplied the resistance genes. Now a virus infect this bacteria. The virus infecting the bacteria are called, yes, bacteriophages. So bacteriophage came, it infect this, and now bacteriophage takes up the resistance gene. Now this bacteriophage multiplies in this bacteria and then it leaves the cell and it infect the another bacteria. So this bacteriophage now infected the second bacteria and send the resistance gene in second bacteria. So the second bacteria also become resistant. So this method of transfer through the bacteriophage is called transduction. Clear? So conjugation was the most common mode of transfer of drug resistance. Transduction is less important. Then the third one, transformation. What happens in transformation is this bacteria which has a resistance gene, by mistake it loses its gene in the environment. So this is free environmental DNA, and this bacteria, it was roaming about and it finds that the free DNA is present, so it will take up that DNA and it also become resistant. So when the resistance gene is transmitted through free environmental DNA, it is called as transformation. So you can assume that एक आदमी घूम रहा था and he found the 2000 rupees note on the road and he picks up that note. So 2000 रुपए का नोट रोज रोज तो मिलता नहीं. So this is the least common method of transfer of drug resistance. So most common is conjugation, least common is transformation. Clear? So that is about the transfer of drug resistance.